Finally, we talk about nucleic acids. Now, nucleic acids have a very different function from carbohydrates, lipids, and proteins. Carbohydrates, lipids, and proteins are used as functional units throughout our entire body. Carbohydrate for quick energy, lipids for stored energy, our phospholipid membrane, our steroids, our hormones. They're constantly working. Proteins are doing all the other work, and they're actually working to break down for that quick energy with carbs and that stored energy with phospholipids. DNA has a more passive role, I guess I would say. DNA is what codes for proteins, by the way. So you have DNA in all your cells except for up here, because these are dead. So not the dead cells on the surface of your skin, but all the other living cells in your body have DNA, and it should be the exact same DNA for each individual cell of your body. The DNA codes for the proteins that work in your body. The reason you have the eyes that you do, the hair, the ears, the reason your face looks a certain way, the reason you're tall or short, some of the reasoning behind whether you're super skinny or a larger person, all of this is by the expression of proteins. The proteins are coded for in the DNA. DNA is information. That's the best way to think about it. But nucleic acids aren't just DNA. I should make that statement clear. It's DNA and RNA. So when we get to nucleic acids, we have the carbs from before, the protein from before, the lipids not so much from before. It's in the nucleus of our eukaryotic animal cells, and if we were a plant, it's in the nucleus of the plant cells as well. Nucleic acids, DNA is the main one. Then you have RNA as well. I believe I told y'all previously, the central dogma, the universal truth, the thing that we know is true for all living things that we have found on this planet so far, is that in order to express protein, like we do, it has to go from DNA to RNA to protein. Which means if you are a virus or a bacteria that only has RNA, you have to hijack a host to get DNA made to make RNA to make protein. So DNA and RNA are our nucleic acids, and these are information storage. It's our information, okay? The function, I'm going to turn down the sound. The function is series of bases encoding information. The bases are adenine, cytosine, thymine, and guanine, A, T, G, C. It's like the letters of a book. It's only 4 letters, right? But the combination that you put them in and the order that you put them in reads differently. So if you were GCCCTA and somebody else was GGTAC or whatever, their proteins are going to be different. The stored information is what we used for our own growth, right? Because we start out as 6 to 10 pound individuals, and you grow up to be whatever you are now. You're much taller than you were when you were born. We grow. So in order to grow, we have to we expand the number of cells that we have. In order to make more cells, you have to copy the DNA and split the cells and have a copy of the DNA in both the new cells over and over and over again for growth. So stored information is passed from cell to cell in growth and from parent to offspring for reproduction purposes. The mother gives half of the 46 chromosomes, so 23 chromosomes in the ovum. The father gives half of the 46 chromosomes, so 23 chromosomes in the nucleus of the sperm. When the sperm burrows into the ovum, the nucleus head of the sperm disintegrates, and the genetic information joins with that of the ovum to form a complete cell. And from that one single cell entity, you get a whole entire human being. So that means it needs to copy accurately for it to happen. The stored information is called genes. So each individual protein that you produce comes from a single gene. And the genes are made up of all the nucleotides, the A, T, G, C. It's genetic information. It's important that we pass on information. Why is it important? There's really no other way to create the template of a human being. You have to have the DNA template to code for the proteins to form the human body to function. So it's genetic material. So these here are chromosomes. This is a karyotype, is what it's called. It's when you take the chromosomes out of a nucleus of a cell, which we can do with our blood cells and we can do with our saliva and whatever, and you go through a whole bunch of processing to disrupt the nucleus and disrupt the cells to only get the DNA. This is condensed DNA. So DNA in the nucleus during a certain phase called interphase is loose and free, so it can be copied. But for mitosis, it condenses down into what we call chromosomes. These are stained chromosomes to show certain bands, and karyotyping is used to see if there are genetic abnormalities or anomalies in a person or a human baby, a fetus. It stores information in genes, and it's the blueprint for building proteins, as I've been saying. Here is that central dogma of DNA. Dogma, D-O-G-M-A, means a universal truth. Once again, nothing can be 100%, but this is as close to 100% as we're willing to go. Central dogma. That DNA has to be coded, it has to be translated. No, transcribed. There we go, because DNA and RNA are both nucleic acids, so it's the same language. So DNA has to be transcribed into RNA, but RNA has to be translated into proteins. RNA is nucleic acid, proteins are an amino acid. It's a language change there, so it's a translation. So there's DNA to protein. It transfers information as being a blueprint for new cells and a blueprint for the next generation. And oh, this is a picture of sperm reaching an ovum. Only one sperm is allowed to penetrate the ovum. There's actually a chemical reaction that happens the second that first sperm penetrates through, so that no other sperm can also enter. Because what would happen if two sperm entered one ovum? If two sperm entered one ovum, then you wouldn't just have 46 chromosomes, you'd have 46 plus 23. You'd have 69 chromosomes in that cell. That would most likely be an unviable offspring, and that pregnancy wouldn't even start. The cell would terminate before it even begin, because you can't have a 69 chromosomal human being. We only have 46. So here is a very lovely slide depicting a chromosome as it is condensed in the nucleus. You can see the X here. And if you pull it out of the nucleus in its condensed chromosome form, you can see this banding that is done, and these are depicting specific genes. If we were to unravel the chromosome into chromatin, which is loose DNA, here you can see the individual bases creating that double helix ladder structure that will code for information in each individual gene and all of the chromosomes. Since DNA is more complicated, we would say, than carbohydrates, lipids, and proteins, we have to actually take the time to talk about the individual pieces of DNA and RNA. So you have RNA, ribonucleic acid. It's a single helix. It's half of the DNA. It's just half. It's a single helix. DNA is deoxyribonucleic acid, and it is a double helix. The structure is made from nucleotides. RNA and DNA share 3 of the same nucleotides, adenine, guanine, and cytosine. But where thymine would be in DNA, uracil is in RNA. So RNA is a single nucleotide chain. Here it is depicted in more of a cartoon form. DNA is a double nucleotide chain. The nitrogen bases, the adenine, guanine, cytosine, thymine, bond in pairs across the double helix change. And you see that it's noted here that the bonding in this center structure is by hydrogen bonds. It spiraled into a double helix, beautiful pictures, that was first proposed as a structure of DNA in 1953 by James Watson and Francis Crick. If you ever have a chance to go read their publication, it is a single-paged publication that is simply written, denoting the structure of the double-stranded DNA. It's pretty much a work of art. In the very end of their paper, they write the sentence, and I'm totally paraphrasing here, that it has not escaped our notice that the structure of the double helix allows for an easy mechanism for copying to occur, which is basically the mic drop of scientific publications. Now, of course, it comes out years later that Watson and Crick kind of stole a lot of information to figure out their model, and they were not the only ones published at the same time. They just published first. And that's kind of the way it works in science. If you're the first, you get all the credit. In 2003, they celebrated their 50th anniversary of this discovery. It's 2020 now, but that's not even 20 years later. So if you think about that for a minute, that means it's only been 70 some odd years, close plus or minus, since the discovery of the structure of DNA. And if you think about how much scientific innovations have occurred in the last 70 years since this, all the genetic manipulation we can do, we can make test tube babies, we can attack cancer, and almost directly we can eradicate all of your cells and put somebody else's cells in there. A lot of this wouldn't have been possible without this discovery. Okay, so what is a nucleotide? It has 3 parts. Follow along. It has a phosphate group. That's not where they're starting. They're starting on the other end. It has a nitrogenous base. So this yellow hexagon here, or the pentagon hexagon here, those are called purines. The bigger structure has the smaller name. These are purines, and these are pyrimidines. We'll talk about more of that in a second. So it has a nitrogenous base. It means it has a carbon-nitrogen ring. It has a pentose sugar, so ATGC for DNA or ACGU for RNA. A pentose sugar, so a 5-carbon sugar. Remember, each point 1, 2, 3, 4, actually I started the wrong way, I'm so sorry. 1, 2, 3, 4, 5 carbons. That 5th carbon is coming off of the 4th carbon, but each point is a carbon here. And then, well, I guess that's important. The pentose sugar in RNA is a ribose, and in DNA is a deoxyribose. So that deoxy should tell you that it loses in oxygen. Look here at the number 2 carbon in RNA, has an OH group coming off of it. Look here at the number 2 carbon in DNA, it only has a hydrogen coming off of it. So we have lost the oxygen in deoxyribose nucleic acid and ribose nucleic acid. And then it has a phosphate group. Phosphate groups typically are used for energy. Are nucleic acids charged molecules? Yes, due to the negative charges on the phosphate group. So, purines and pyrimidines, those are the different types of nitrogen bases. Your purines have that double ring nitrogen base, the pentose ring and then the hexose ring bound to it. This is your adenine and guanine. The pyrimidines have a single ring nitrogen base, cytosine and thymine. A purine always binds to a pyrimidine through the hydrogen bonds. If you had purine-purine binding together, that would be too wide. And if you have pyrimidine-pyrimidine binding together, that would be too narrow. And you would have a structure that, you know, widens and narrows as you move up and down it. It wouldn't function properly. So a purine has to bind to a pyrimidine. That also includes uracil. If you are well versed in the periodic table, knowing that Ag is silver, pure silver. Ag is a purine. It's kind of a mnemonic if you want it. To build the polymer, every single thing is going to have the same ribose sugar, either it's RNA or DNA, and the same phosphate group. And so look here, please. You have a phosphate sugar, phosphate sugar, phosphate sugar, phosphate sugar, phosphate sugar, phosphate sugar. I'm not lying to you when we call that the phosphate sugar backbone of the DNA. Then, depending on what nitrogenous base is sticking out here, you're going to want to find the partner opposite base. So let's say this is adenine, and it's a purine, then you want thymine pyrimidine on the other side. If this is a cytosine pyrimidine, you want guanine purine on the other side. I always think the bigger, no, I'm not even going to say that because then you'll think that too. The sugar to phospholipid or phosphate bond is called a phosphodiester bond. That's the fourth one. Whoops, not 5. That's the fourth one. Wait, I made myself small so you can't see me. That's the fourth one. The first one was in carbohydrates, and that's the glycosidic linkage. The second one was in lipids, and that's an ester linkage. The third one was in proteins, and that's a peptide bond between the amino acids. Here we are in nucleic acids, and it is a phosphodiester bond. All of them are formed by dehydration synthesis, losing of water. They just have a different name depending on the biological macromolecule that they are. You do need to know those differences. So this is a phosphodiester bond. This is a phosphodiester bond. This is a phosphodiester bond. Every time that sugar is bonded to a phosphate, phosphodiester bond. A new base is added to a sugar of the previous base. The polymer only grows in one direction. And when we get to the end of this semester, when we talk about DNA replication, transcription, and translation, you will have to know about the directionality of growing DNA. The nitrogenous bases hang off of the sugar phosphate backbone. Dangling bases allow for bonding of the other bases. Whee! There we go. Nucleotides bond between the DNA strand. If you are adenine thymine, you have 2 hydrogen bonds. If you are guanine and cytosine, you have 3 hydrogen bonds. I've already mentioned why matching the bases are important. It's due to the size of the structure. This allows for equal sizing all the way across the DNA. 2 pyrimidines would be too small, 2 purines would be too big. That is actually one of the reasons it took Watson and Crick so long to figure it out. They also had, at one version of their molecule, they had the bases facing outward, which we can laugh about that now, but then, you know, they were searching in the dark. They didn't know what they were looking for. They were just trying. So double helix, these hydrogen bonds between the bases join the 2 strand. Remember, A and T have 2 hydrogen bonds. C and G have 3 hydrogen bonds. Why do you think it's important that the bases are joined by our weakest form of bonds, hydrogen bonds? I'm actually going to give you more time to think about it because a bell is about to ring overhead and it's going to be disruptive if I'm talking during it. So why hydrogen bonds to hold the bases together? Okay, the reason being, your time is up. You have to be able to easily break them apart so that you can copy the DNA templates. To copy DNA, you have to understand that the 2 strands of DNA helix are complementary, which you do understand. If you know that a G is here, then a C is going to go here. If an A is here, a T is going to go there. It's complementary, which means that even if you pull it apart, only if you read one side, you know exactly what should be on the other side. So if you have one, you can build the other. And if you have one, you can rebuild the entire structure. That's the basis for the replication of DNA. This is a good system because it works. When in the life of a cell does DNA have to be copied? Excuse me. It has to happen in cell reproduction during mitosis. This is the end stage of mitosis where two cells are splitting apart. All the other time in between, the DNA is loose and open in the cell for life processes to occur. This is called interphase. During mitosis, all the chromosomes condense, so you can't make new proteins off of it, and they, come together and then pull apart. It's called PMAT, prophase, anaphase, metaphase, telophase, and then you have cytokinesis to separate the two cells. So that's mitosis, or in gamete production, to produce ovum in the ovaries, which happens for females. We actually have all of our ovums complete before you're even born, and to produce sperm in the testes of men, which happens daily after puberty. And here is that sentence. Here is that beautiful sentence that ended their paper. It has not escaped our notice that the specific pairing we have postulated immediately suggests a possible copying mechanism for the genetic material. I mean, they probably blew some minds with that sentence. Well, they blew minds with their entire paper. Once again, you should read the paper. I forget what publication it was in, but you can find it very easily. And so if you split apart the DNA, if you break all those hydrogen bonds, which we know breaking hydrogen bonds is easy individually because they're weak bonds, then you can separate the templates and build new template strands or new daughter strands along with the original template, and you get two identical copies of DNA to separate into the cells or to separate into sperm or whatever. So Watson and Crick were the main players. There were others involved. We are going to watch a documentary. It's a short it's more of a short film over their discovery further on in this semester. Maurice Watkins and Rosalind Franklin. Rosalind Franklin found it out. This is a crystallography of DNA. It's a type of X-ray that she performed on DNA. Unfortunately for Rosalind Franklin, she didn't know what she was looking at, but Watson and Crick did, and they actually stole this from her study. But this is showing you this cross in the middle. Those are all the nitrogenous bases. So it's showing you that the bases are in the inside, and it's showing you that they're equidistant apart. And this is the phosphate backbone, phosphate sugar backbone. And this X-ray crystallography is what allowed Watson and Crick to finally understand the main thing that they were missing in forming their structure. Rosalind Franklin died in 1958, very young, from cancer, from performing X-ray crystallographies. And she was never acknowledged by Watson or Crick in their discovery. It came out years later, of course. And I think she was posthumously rewarded, but I'm not really sure. The interesting note, the ratio of A to T and G to C affects stability of the molecule, because if you have more A to T's, then you're losing hydrogen bonds. And if you have more G's and C's, then you have more hydrogen bonds. So in biotech procedures, they have more G and C. They need a higher temperature to separate the strands. If they have a high temperature organisms, which we have a lot of organisms on this planet that can live in volcanoes and steam vents of steam, whatever they're called, and geysers and all that, they'll have many guanine cytosine pairs. Parasites have many A and T, but we don't know why. Another thing to note is that this is the nucleotide adenine. And if you add on 2 more phosphates, then it's our energy molecule ATP, which we use in the body. Not only add ATP, but we also have CTP, TTP, and GTP. So it's a modified nucleotide. Here's your nitrogenous base. Here's your ribose pentose sugar. Here is 1 phosphate, and then they add another and another. The cleaving off of these phosphates phosphates are super high energy molecules because of all the oxygens that are attached to them. When you cleave off one, you release a lot of energy. Here is a beautiful structure of a double helix. It's a model, I don't know where, and here's a person hanging out inside it just to show you for scale. That's it for nucleic acids and carbs and lipids and proteins. So let's do a quick review. Carbohydrates, their monomer is a monosaccharide. The function is for energy, raw materials, energy storage, and structural components. Remember, starch and cellulose and chitin. They are formed through dehydration synthesis of glycosidic bonds. And examples are the polymers, which are polysaccharides, are glucose, starch, cellulose, and glycogens. Lipids don't make polymers. Their building blocks are fatty acids, cholesterol chains, or cholesterol rings, glycerol in long hydrocarbon chains. Their function is for energy storage. Remember, at least is 2 times the amount of energy of carbohydrate, but it takes more energy to break those bonds. So it takes energy to get the energy. They're in our membranes as phospholipids, and they are our hormones and steroids. So fat, phospholipids, and steroids. They are formed by ester bonds or ester linkages between a hydroxyl group and a carboxyl group through a dehydration synthesis. Proteins, their monomer is amino acids, and then they have levels of structure. Remember, they have the primary level, which is just the chains of amino acids that are formed together by dehydration synthesis between the amine group or the carboxyl group of one end and the amine group of another, and they make a peptide bond. The secondary structure is alpha helices and beta pleated sheets within that linear structure. The tertiary structure is when there are hydrophobic interactions to the inside, so they keep the hydrophobic regions internal, and disulfide bridges and ionic bonds between the side chains all along the outsides. It stabilizes the structure. That is the end for some proteins, but for others, you have a quaternary structure, or for most, you have a quaternary structure, which is multiple polypeptides together functioning in whatever they're supposed to do. The function is enzymes, transport signals, defense, structure, receptors, name it, a protein is involved. Examples are digestive enzymes, membrane channels, insulin, hormone, and actin, among all the many other things. Nucleic acids, the structure is the nucleotide, the monomer is the nucleotide. The function is for information storage and information transfer, and the examples are DNA and RNA. You form phosphodiester bonds through dehydration synthesis between a hydroxyl group off of a sugar, a ribose sugar, and a hydroxyl group off of a phosphate. And that is a dehydration synthesis that forms a phosphodiester linkage. So, as the sperm would say to, I don't know, Yoko Ono, I don't know who that is, let's build some DNA, baby. Thank you so much for listening to these 4 lectures. If you have any questions, feel free to email me in mills-more@irvingisd.net.